Okay, so why are hydrogen bonds so important? Not just water is capable of hydrogen bonding, but many, many biochemical particles do this. In fact, most of the chemistry in your body works based on hydrogen bonding. So, for example, in DNA, when DNA, by the way, if you don't know, DNA is basically what gives the blueprint for how your body should be constructed. It tells your body how to make different kinds of cells, different kinds of proteins, different sort of components to run your body. All right, and so we think of it as information storage. And the information is stored in such a way that you get actually 2 copies for every piece of DNA. So this is an animated drawing. If you go pull up the lecture notes, you'll see that this will spin around and show you what a real piece of DNA looks like. This is what we call a crystal structure. So it's almost as if you used X-rays to take a picture. On the particle level, you can actually see how things are connected. So this is a nice way of visualizing it. And one of the most interesting things is that the 2 strands that make up your DNA never actually touch each other in the middle. So if you look at this spot right here, it's a really good view. This is looking straight at 2 of the DNA bases, and there's a gap there. They're not touching. They're not bonded. Unlike all of the connections along the side here, that's all bonded atoms. But in the middle, where the 2 bases come together, there's no bond. It's just kind of floating there together. And that's true for every level of this helix. It's hard to see in some spaces. You can see it right here too because of the angle. But when you look at this in animated form online, you'll see that none of those bases in the middle touch. You have big gaps at every single one of these rungs of the ladder. So those gaps are hydrogen bonds. And so here's an example. Here's guanine and cytosine, G and C. And they pair up perfectly because the shape of each one of them is kind of complementary to the other side. We call them complementary base pairs. So for example, this NH is very capable of forming a hydrogen bond because we said anything with NH can be a hydrogen bond. In order to do that, it's got to have somewhere with kind of like a negative charge. So the hydrogen is partially positive. The nitrogen is partially negative because it has those unpaired electrons on it. You could draw the Lewis structure of that if you can't recall what they look like, but nitrogen almost always has an unpaired pair of electrons. Oxygen really likes electrons, so that's a negative charge there. So what we're getting is a partially positive and a partially negative affiliating with each other. So that's a kind of dipole-dipole interaction. But it's a really strong one because nitrogen is very electronegative. So are oxygen and fluorine, which is why these are the only ones capable of hydrogen bonding. Carbon, which is right next to nitrogen, has a value too close to hydrogen in order to create a dipole that's this strong. So I want you to go look at the electronegativity table in your textbook. Let me find out where it is. Hang on. Okay, so what you've got to do is go look at page 195 in your textbook, and I want you to figure out what the electronegativity difference is between NH, OH, FH, and also look at CH. So CH is not a hydrogen bond, but the other 3 are. So that comparing like this will allow us to figure out what's the cutoff. Why is nitrogen so special? And things like carbon and phosphorus don't do hydrogen bonding. Okay, so go ahead and take a look at that and write down your findings here in your notes so I can see it in class. Also be sure that you check out the animated DNA sequence here because it's pretty cool to see that the bonds actually don't touch. Okay, so on this side we have NH here, so that's a hydrogen bond donor, and it's going to donate into this nitrogen. So you've got positive and negative again. They're sort of flipped from the last one. The hydrogen bond donor is on the left and the acceptor is on the right. Acceptors are always going to be negatively charged, so down here we can apply the same logic. Okay. So you have to have a hydrogen bond donor and a hydrogen bond acceptor in order for hydrogen bonding to work. Okay, so when ice freezes, the way that it freezes is based on the intermolecular forces that water has. And water always freezes into a hexagon. We call this, of course, hexagonal geometry. And so the fact that water freezes into a hexagon gives us the shape of snowflakes. Okay, so go into the lecture notes and click on this link and watch how snowflakes form. This is just an example, but it's all about intermolecular forces, okay? And one of the interesting things we'll notice is that the intramolecular force that holds water together, hydrogen and oxygen together in water, is pretty short. It's 1 angstrom, which is 1 angstrom. Angstrom has a funny symbol. It's like an A with a little degree over it. 1 angstrom is 10 to the negative 9 meters, essentially. All right, so it's very small. So a bond is one of those, but an intermolecular hydrogen bond, so IMF here, is longer. It's almost double. It's 1.8 angstroms. Okay, so bonds are always closer together, which means they're stronger. They have more energy in them. Intermolecular forces are weaker. Hydrogen bonds are one of the strongest kinds of intermolecular force. The only one that's stronger is called ion dipole. And so down here, an ion dipole is exactly what it sounds like. You have an ion. Let's say in this case, we'll look at sodium ion. If I put sodium ion in water, we know that our water molecule is going to be polar. So we have oxygen, and then we have hydrogen. So that means that the dipole for that is going to go kind of straight up through the middle, because the oxygen is negatively charged and the hydrogens are positive. So if I have an Na+, then what's going to happen is that the oxygen is negatively charged in water, so it's going to want to be as close to the positive charge as it can get. And then the hydrogens of water are going to kind of stick out the back. So this is the dipole pointing towards the positive charge because the arrow end of it is negative. And so you'd get a whole bunch of water surrounding the sodium like that. So this is basically the last intermolecular force. It can be pretty strong, but it kind of depends on what the two things being mixed are. So if you have a solution, so you have water mixed with an ionic compound, that's a pretty strong interaction, okay? But don't confuse it with the compound by itself. The compound, sodium chloride, is held together with ionic bonds, which is much stronger than any intermolecular force would be. Okay, so let's look at this figure. This is from another textbook, but I really like how this figure lays out the different intermolecular forces. So we said that dispersion is the weakest. So dispersion can have around all the way from like basically 0 kilojoules per mole of energy all the way up to about like 4 or 5. So we can, dispersion is like about 0 to 5 maybe kilojoules. Dipole is the next strongest, and it ranges from around 2 to maybe 12 or 13 kilojoules per mole. So our units are down here. Hydrogen bonding is next, and it ranges from about 15 to 25 kilojoules per mole. And ion dipole kind of overlaps with that. Sometimes it can be kind of weak. It can kind of line up with your dipole. That depends on what combination of materials you have. But so ion dipole ranges from about 12 all the way up to quite high. Okay, so we're looking at maybe, I don't know, 60 kilojoules per mole. I'm guessing. It's the axes aren't very particularly precise there, but So that's a pretty big range for ion dipole. It is sometimes a little tricky to rank hydrogen bonding or ion dipole as being which one is stronger than the other. So those can be a little fuzzy. But the others are pretty obvious. Dispersion is always the weakest. Dipole is a little stronger than that. And both hydrogen bonding and ion dipole are stronger than that. So it kind of creates a continuum here. And then there's kind of a big gap. And we go quite a long ways until you get to ionic bonding, which they've marked here as ion-ion. And ionic bonding can range from somewhere around 200 kilojoules per mole all the way, all the way up to almost anything, but we'll say 2,000 kilojoules. Covalent bonds kind of overlap with that, around 200 kilojoules, like that. But we think of covalent bonds as being pretty strong. This 2,000 kilojoules would be a pretty rare instance for ionic bonding. But this, let's say it goes to like 1,000. We're talking about individual bonds, and covalent bonds are usually made up of multiple bonds. So covalent compounds tend to sort of add up quickly. All right, but this is a general idea for how to rank the different strengths of intermolecular forces. The bigger the energy, the stronger the force. And the stronger the force, the more likely something is to be a solid, because solid particles are as close together as possible. So what we want to do here is predict what the dominant intermolecular forces are for each thing. We know that everything has London dispersion forces. So if I can't identify any of the other three, then that means LDF is what we have. And the process here is that I need a Lewis structure. I need to find the shape of the molecule. And then I need to find the polarity of the molecule. Once I've done that, I can decide whether it's going to be which IMF, which of the four. So CH4 has a carbon in the middle and 4 hydrogens attached. It doesn't have any unpaired electrons, so that means that this has a tetrahedral shape, both in the electrons and in the molecule. And it is non-polar because tetrahedrons are symmetric. Okay. If it's non-polar, we said the only thing that has that works, the dominant force that works for non-polar substances is always London dispersion forces. Okay, so this one is London dispersion forces. We're going to do that same essential analysis for every single one of these. CCl4 would be carbon in the center and 4 chlorine around it. I do not want to forget all of my little electrons, so we put them in. Okay, so there's our structure. Again, there's 4 domains. None of them are bonding. Or sorry, that was wrong. None of them are non-bonding, so that gives us a tetrahedral geometry. We said tetrahedrons are always symmetric when the atoms are the same on each branch of the tetrahedron. So that means this would be a non-polar substance, which means, again, London dispersion forces. Now, in deciding which of these two things is sort of stronger LDF, what we need to do is think about electronegativity differences. Okay, so London dispersion forces relies on the idea that the electrons are sloshing around. Okay, so we want to figure out which one of these two has electrons that are, like more sloshy than the others. I made up a word for you. I don't even know what sloshy means, but you get the idea. So we can figure out how sloshy electrons are by subtracting the electronegativity values for one of the bonds. So carbon and hydrogen would be 2.5 minus 2.1. So we get an electronegativity difference of 0.4. Carbon and chlorine would be, let's see, chlorine is 3.0 and carbon is 2.5. So whichever one of these has the bigger difference in electronegativity is the one that is, sloshier. So that means carbon tetrachloride, CCl4, would have stronger London dispersion forces. So if I had to rank these two, I would choose CH4 as the weakest one, so most likely to be a gas, and CCl4 would be the one most likely to be a liquid.